Hi, welcome to the Brain Initiative workshop on dissemination of non-invasive imaging technologies. I'm John Ngai, the director of the NIH Brain Initiative, and I'm very much looking forward to an exciting 2 days of science and engineering in this workshop. I just wanted to give you a background of the Brain Initiative to put your work and our work into a larger context. Now, as many of you might know, the Brain Initiative, or the Brain Research Through Advancing Innovative Neurotechnologies Initiative, has a mission to revolutionize our understanding of the human brain by accelerating the development and application of new and innovative technologies. This was announced by the White House in 2013, with the first awards being made in 2014, and represents a partnership between 5 U.S. federal agencies and private foundations. Now, the goal of the NIH Brain Initiative is to develop and apply new tools for understanding how neural circuits underlie complex behaviors in models of health and disease. To achieve these goals, I'd like to keep our sights set on the following 3 issues. One is to leverage the technological innovations that will allow us to make new discoveries about neural circuit function. And the second then is to use these discoveries as a foundation for new therapies for human brain disorders. And third, and germane for the topic of the next 2 days of workshop, is to disseminate and democratize these technologies for basic discovery and clinical applications. Now, as I mentioned, the Brain Initiative started late 2013, 2014, and I think it's fair to say that much of the success we've enjoyed so far, with a lot of hard work on everyone's part, can be traced back to a very well thought out and well-written strategic plan by a group that was commissioned by NIH Director Francis Collins. This group was led by Professors Bill Newsom and Corey Bargman, and this group came up with a set of recommendations for how to proceed on this remarkably exciting initiative based around 7 main areas. The first was to discover the diversity of cell types that comprise the brain and ultimately of the human brain. The next is to map the connections, these circuit diagrams in the brain in multiple scales, and to monitor neural activity that runs through these circuits. And I think many of you here are directly involved in some of these efforts. And then next is to devise and implement precise interventional tools for both perturbing as well as modulating neural circuit function. And this will help us demonstrate causality, but also provide tools for intervening in human brain disorders. And then the next few points are really about having what we do be solidly grounded in theory and data to advance these discoveries into understanding how the human nervous system works, and to integrate all of the above approaches. So this was the report that was laid out for us at the beginning of the initiative. As with any well-conceived and well-run initiative, there was a very careful look again at our progress by the so-called Brain Working Group 2.0. There were actually 2 groups, one focusing on neuroscience, the other on neuroethics. And this was a group, 2 groups actually, led by Kathy Dulac and John Mansell, as well as Jim Eberwein and Jeff Kahn, focusing on the neuroethics component. And this was a chance to reflect on what our progress had been made in the first 5 years or so, and what should we continue investing in, and what directions might we wish to go into in the future. So there were 4 main takeaways from these 2 reports. The first was to stay on the productive path already underway. We've all seen a lot of great progress in these areas, both in terms of technology as well as discovery. We certainly want to continue along those very productive lines. The second was to be careful to balance individual investigator research with team science. There's a place for both in the enterprise. We need great questions to drive the development of great tools, and we need great tools to drive the next set of great questions. And both are really vital for advancing our understanding of how the brain works. The third was to devote ample resources to scale up large projects. So we can look back at the last 5 or 6 years and see a remarkable set of resources that we have generated, not only within the Brain Initiative, but in fields related to us, other fields of biology, genetics, and very importantly, in the engineering and physical sciences. So now might be a good time to invest in large projects that could truly transform the way we conduct our research. And then finally, we really are working very hard to enhance the integration of neuroethics with neuroscience and to support the high impact work that really does consider what are the broader societal implications of all that we do. So I'd like to focus just for a moment on this idea of this being a great time to stand up large projects that could transform the way we conduct neuroscience research and ultimately benefit the human condition. And through a series of internal workshopping and taking the input and advice from many, many different groups, including through the Brain 2.0 working groups, the Brain Initiative is embarking on this next phase, the so-called Brain 2.0 transformative projects that we feel will transform the way we conduct our research. And these three large projects that I'll describe to you just very briefly build on a solid foundation of discovery and technological innovations. The first project that I'll talk about just very briefly is the phase 3 of the Brain Cell Census Project, or the so-called parts list of what comprises the brain. It builds on the success of the Brain Initiative Cell Census Network, or BICCN, which is focused primarily but not exclusively on creating this parts list of the mouse brain. And this is not just based on molecular data, but really focused on integrating molecular with physiological, morphological, and connectivity data to give us a multimodal, comprehensive view of what cell types comprise a mammalian brain. For this next phase, and largely based on the success of this mouse-centric project, we will be shifting our emphasis to construct a comprehensive human brain cell atlas, and we expect to launch this project in the coming year or so. The second is this development of next-generation technologies for brain microconnectivity analysis, or the wiring diagram. Now, many of you here are studying the wiring diagram of brains at different levels, and we feel that understanding how this marvelous biological computer works will require not only a parts list, an understanding of the parts that go into it, but how they connect to each other. And ultimately, we would like to do this for a whole brain at synapse level resolution. And this will require the identification and validation and scaling up of new technologies to handle not only the incredible load for data acquisition, but also for data analysis. So the third large project is the so-called armamentarium for gaining access to cell types. Many of us use rodents as a model system for studying the nervous system. And because, among other reasons, we are afforded great access to cell types through genetics, namely through transgenesis. This, of course, isn't possible for a number of reasons or feasible or practical in other species, including non-human primates or humans, certainly. And so we need better ways of accessing cell types through non-transgenic means, either using viral vectors or nanoparticle chemical vector delivery systems. And so our goal here is to develop a large system, a large toolbox, if you will, for accessing cell types across a number of species. The long-term goal is to develop tools that will allow interventions into human brain disorders through either molecular or gene therapies. Now, this will really both enable as well as take advantage of the output from these first two projects. The first two projects give us a ground truth for how the nervous system will work, and this third large project will give us a way for getting in there to characterize these circuits, for modulating them, and ultimately for devising new cures. And I'm very excited that our first RFA has been issued with a receipt date coming this February. So this is the broad strokes version of where we see brain heading in the next 5+ years. I think it's fair to say that the discoveries supported so far by brain lay the foundation for devising new interventions in human brain disorders. I think it's also very fair, or more than fair, to say that brain-supported non-invasive imaging technologies, many of which you folks have developed here, are unraveling the circuits and computations underlying complex behaviors, including human behaviors. And this will allow us to identify in great detail the cells and circuits affected in neurologic and neuropsychiatric disorders in humans. And this really highlights the importance of technology dissemination to enable and accelerate knowledge generation and development of new therapeutics. So I'd just like to take a few minutes here and to talk about some studies that have been done on humans. And here I'm not going to focus on studies using non-invasive imaging technologies, but other studies that highlight perhaps how we might imagine how imaging technologies might dovetail with these to give us a greater understanding of how the brain functions in humans and ultimately how we might treat human diseases. So here's a very interesting study by Dr. Nantya Sudana and her colleagues at UCLA. What they've done is they've used a bidirectional electrode that's already been implanted in patients to control epilepsy. And they've used this as a recording device, and they've were able to acquire data from this device in a backpack that the subject literally can wear and walk around the room in, while they simultaneously obtain other data on that subject, including their movements, heart rate, skin conductance, that reflects perspiration, respiration, eye tracking, scalp EEG, as well as their position, their global positioning in the room. And you can see this in a movie on the left. And they've made some really cool discoveries. And one is that there's a high theta band power in the medial temporal lobe that becomes active when subjects navigate to the boundary of the room. And what's really quite interesting is that one can see a similar activity when the subjects watch another person navigating to that same place in the room. So there's both a representation of self and other. I think studies like this really will give great insights, given that we can now study on a limited basis, of course, the correlation between neural activity and behavior, movement, etc., in a freely moving, awake, behaving human. So this is really quite exciting for the future. Here's a study from Aysel Gundoz's lab at University of Florida, just published at the end of last year in Science Translational Medicine, where they're using an adaptive deep brain stimulator to control essential tremor. So here's a patient, a subject who has clear issues with controlling, pouring this cup of water from one to the next. And then what the stimulator does is it senses activity, turns on the stimulator, and then is able to control this tremor. Really quite remarkable in many, many ways. And I think this points to the ability to identify circuits that are involved in various disorders and to control them with these adaptive stimulator devices. And I think the more that we understand about circuits through imaging and other technologies, the further along we can advance these technologies. And finally, perhaps known to many of you here is the work of Dr. Eddie Chang, who has used recordings in interoperative patients, the ECOG recordings, to record the activity in the part of the brain involved in speech generation, and use this combined with kinematics of this actual speech generation in the musculature, in the jaw and in the mouth, to create a speech synthesizer based on a very involved recurrent neural network, two-stage neural network encoder. So this really provides a number of interesting possibilities. First is to understand the neural basis of speech generation, but also to generate potentials for speech prosthetics in patients who are unable to speak because of either, for example, ALS or due to stroke. And I really think that this combined application of neurophysiology with very detailed kinematics with these very intricate machine learning algorithms really speaks to the future being almost limitless in terms of what we can do with good data if we know what to do with it. So let me just close with a final few thoughts about what we're doing. And as you can see, I've highlighted 3 case studies involving humans as research subjects. And with this in mind, we need to be quite mindful of not only what the benefits of our work might be, but also what are the risks. And risks not only to the human subjects, but also to the way the field is going to be going in general. So Brain has been quite proactive in this regard. We have a standing neuroethics working group. Again, much of what we do has been guided by previous strategic plans by the working groups commissioned by Dr. Francis Collins. We have a set of neuroethics guiding principles that cover, of course, issues regarding safety and privacy, but also since we're literally dealing with the human brain and therefore the human mind, our personalities, we need to be very careful about issues of agency, malign and dual use. I mean, many of the tools we're developing here may be initially aimed at treating human brain disorders, but they can also be used for enhancements. And of course, we need to very carefully engage the public and democratize what we do for the benefit of all. And finally, I just want to touch on Brain's role in training the next generation of scientific leaders. And our view is we need diverse perspectives and we need new, fresh ideas. Here are a series of programs we have. We have an F-32 program We have a diversity-targeted Brain K99 R00 program where these awards are open for people of underrepresented minorities as well as to women. And in a related set of awards, we have a Blueprint D-SPAN award targeted now for late-stage graduate students and early-stage postdocs. And one thing I'd like to draw your attention to are these Brain Initiative diversity supplements. Anyone here with a active brain award can apply for funds to support a student or a trainee from an underrepresented background. And I direct you to the Brain Initiative website here. And finally, I would like to thank the many, many people, over 100 dedicated staff that make Brain a reality and help you do the great work that you do. And with that, I will thank you very much and look forward to a great workshop.